Welcome to the fascinating world of immunology. Today, we are going to look at antibodies from a completely different perspective than you might be used to. Usually, we think of antibodies as the heroic weapons our body uses to hunt down foreign invaders like viruses and bacteria. But what happens if we take an antibody from a human and inject it into a rabbit? To the rabbit, that human antibody is a foreign invader, and the rabbit's immune system will attack it. Let us dive into how this works. Now that we understand that antibodies can be targets, let us look at why this happens. Because antibodies are large, complex glycoprotein molecules, they are highly immunogenic when introduced into a different species, or even a genetically different individual of the same species. To understand how this works, we can use a simple car analogy. Think of the antibody's general structure as the make and model of a car. Its genetic variations are like a custom paint job. Finally, its unique antigen binding site acts as the highly specific license plate. Building on our car analogy, let us formally define what these parts are. In formal immunological terms, an immunoglobulin antigenic determinant is a specific region on an antibody molecule that can be recognized as an antigen by another immune system. Immunologists categorize these determinants into three major classes based on where they are located on the protein and who shares them. The first class is the isotype. This refers to the constant region determinants that are shared by all individuals of the exact same species, much like the standard factory model of our car. Having established the baseline isotype, we now look at the variations between individuals. The second class of determinants is the allotype. These represent small genetic variations located in the constant regions of the antibody. Unlike isotypes, allotypes differ among individuals within the exact same species. You can think of them as being very much like human blood types or a custom paint job on a specific car model. Because these are genetic markers inherited directly from our parents, they play a crucial role in distinguishing one individual's immune proteins from another's. Now that we understand species-wide and individual variations, we reach the most specific level. The third and final class is the idiotype. This is the completely unique sequence of amino acids found exclusively in the variable region of the antibody. The idiotype is what determines exactly which specific antigen the antibody can bind to. Returning to our car analogy, if the isotype is the car model and the allotype is the paint job, the idiotype is the highly specific license plate. This unique binding site is what makes every single B-cell clone special and capable of fighting a specific disease. Now that we have defined the three classes, let us see them visually on the antibody molecule. Here, you are looking at the classic Y-shaped structure of an immunoglobulin. Notice the red regions at the very tips of the upper arms. These represent the idiotypic determinants, which form the unique antigen binding sites specific to each clone. Moving down, the light blue regions making up the rest of the arms and the main stem represent the constant regions. This is where we find both the isotypic determinants, which define the antibody class, and the allotypic determinants, which carry the subtle genetic variations between individuals. With a clear visual understanding, we must ask why these determinants matter in real life. Understanding them is absolutely critical for modern medicine. For example, if a patient is bitten by a venomous snake, we often give them antivenom made of neutralizing antibodies produced in horses. Because horse antibodies have a different isotype, the human immune system eventually attacks them, causing a dangerous inflammatory condition called serum sickness. Furthermore, recognizing allotypes helps in forensic science and paternity testing, since these markers are inherited. Finally, idiotypes are incredibly useful in treating B-cell cancers, like multiple myeloma, allowing us to design drugs that target only the cancerous cells. Knowing how crucial idiotypes are in clinical settings, let us look at the mathematics of how the body generates so many of them. The unique idiotype is created by randomly mixing and matching different gene segments in the DNA. This formula calculates the baseline combinatorial diversity. The variable I stands for the total number of unique idiotypic combinations. The variables VH, DH, and JH represent the number of variable, diversity, and joining gene segments available for the heavy chain. Similarly, VL and JL represent the segments for the light chain. If any of these genetic variables increases, the total number of unique idiotypes I will increase exponentially, giving the immune system a vastly larger repertoire. Now that we have our combinatorial formula, let us apply it to a specific laboratory mouse model. We are given the exact number of gene segments, 100 variable, 12 diversity, and 4 joining segments for the heavy chain, plus 35 variable and 5 joining segments for the light chain. We need to calculate the total unique idiotypic determinants. First, we multiply the heavy chain segments together to get 4,800 unique heavy chains. Next, we multiply the light chain segments to get 175 unique light chains. Finally, we multiply these two results together. The final answer is 840,000, meaning this mouse can recognize 840,000 different specific antigens before any mutations occur. While the baseline calculation is impressive, it leads us to an even more complex biological reality. The immune system is much smarter and messier than simple combinatorial joining. When gene segments are spliced together, enzymes randomly add or remove nucleotides at the junctions. This process is known as junctional diversity. To account for this, we use an expanded formula. The variable IT represents the final realistic number of unique idiotypic determinants. IB is the combinatorial baseline diversity we just learned to calculate. MJ represents the massive multiplier effect caused by the random splicing process. If this multiplier increases, the true diversity skyrockets, showing the staggering scale of the immune repertoire. Let us see this expanded formula in action with a human patient. We are given a baseline combinatorial diversity of 3 × 10⁶, and a junctional diversity multiplier of 3 × 10⁴. We need to find the true total number of idiotypic determinants. We substitute these scientific values into our equation. To multiply these numbers, we first multiply the base coefficients, meaning 3 × 3 = 9. Next, we add the exponents of the base 10, meaning 6 + 4 = 10. The final answer is 9 × 10⁻10. Physically, this means the patient can produce 90 billion uniquely shaped idiotypic determinants, ready for almost any pathogen on Earth. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that immunoglobulins are complex proteins that can act as antigens themselves when placed in a foreign environment. Second, we explored the 3 levels of structure: isotypic determinants, shared by the whole species; allotypic determinants, representing individual genetic variations; and idiotypic determinants that form the unique antigen binding sites. Finally, we saw how mathematical combinations and junctional diversity give the immune system a nearly infinite repertoire. Understanding these concepts is the absolute foundation for using antibodies in modern medicine and targeted therapies. Great job following along.